In general, the computational heat discovery is a thing that has been around for several decades now, and there's a plethora of strategies to do the computational heat discovery depending on what is known, whether the ligand structure or protein structure is known or unknown. The approach to the heat discovery is different, and of course we start with if it's a good thing if the structure of the protein, target protein, is known, and then of course dock and score and virtual screen can be done fairly well. And then if on the other hand, we don't know the protein structure, but have the biological activity data or some ligand marker, then of course ligand-based approach is more convenient, where pharmacophores, 2D and 3 similarity methods, QCR methods, virtual screening are all applicable. Also now a growing trend is the de novo design, is a modern trend in computational drug discovery. We will talk about it a little later. And of course, if nothing is known and basically we are starting from the very beginning, then the compound libraries are very suitable tool to actually find ideas for future drug discovery programs. And then Enamine compound libraries are very useful tools in this case. So the opportunity space for the finding ideas and heat discovery is huge. It's billion of drug-like molecules, but we also have quite nice data from some already approved drugs or drugs that already are in clinical trials, so we can have those as references. And also on the other hand, we have almost 20,000 human proteins, more than 20,000 human proteins in the body, of which around more than 600,000 are actually 3D profiled. I mean, the crystal structures are available, and around almost 3,000 of them are actually drug targets, which has been researched or somehow tested in screening campaigns. So a lot of methods is used, and nowadays everybody is talking about machine learning and deep learning and what is called artificial intelligence. So in fact, again, a lot of machine learning methods are available for decades now, like, for example, support vector machines, decision trees, key nearest neighbors, etc. All those algorithms has been the handy tools for computational chemists and biologists for quite a lot of time. But since 2012, there is a growing trend for applying deep learning, so-called deep learning, which is kind of advertised and widely regarded as a new paradigm changing tool in computational drug design. So let's try to see why that. First of all, the definition, as deep learning is actually also a machine learning technique, but it is a very specific set of algorithms built on the idea of the how human brain operates. Essentially, it's built on the neural nets, and there is a lot of innovation that has been done around this particular type of architecture. And in 2012, there was a very marked event, the ImageNet competition, where essentially the very good results with image recognition has been achieved on a large-scale data set of images. And that was kind of maybe symbolic, maybe, but that was the event that triggered a lot of investment in the area and a lot of gigantic shift in the perception of how deep learning can also be applied for pharmaceutical research problems. And but if we delve a little bit deeper into actually what's the actual difference, why deep learning is better, or maybe it's not better than the rest of techniques. First of all, much as computational quantity structure activity relationship models are useful and has been useful, but they still have pretty time-consuming stage to separate training set and test set. For example, they have limited, certainly they have limits regarding the size of data to use for. If we talk about hundreds of millions of variables, that's not something that some legacy machine learning methods can handle sometimes. And also types of data and different unstructured data is also a challenge for most of the technologies that are usually regarded as machine learning or statistical methods. In contrast, the deep learning, probably the biggest advantage is that it can learn representation of data by itself. So basically, in machine learning, typically the something like fingerprints or something has to be actually instructed and thought over by actual scientists. Whereas the deep learning architectures allow to essentially learn features automatically, so to speak, and create automatic data representations. And those representations are very complex and not basically understandable by humans in most cases, but they are actually leading to a good prediction and good compound clusterization for classification, for example, or activity prediction, etc. And so deep learning is used and very useful in those cases where we talk about big data, where the screening campaign involves, let's say, really hundreds or tens or hundreds of millions of compounds, or where optimization is to be done on a very large data set. Also, it opened the room for a completely new set of problems that can be now solved. For example, image classification, image recognition, natural language processing, and speech recognition. All those things, they are impossible to do with most of the statistical methods of the legacy school, but in the same time, some of these applications are relevant to pharma, like, for example, analyzing articles, research articles, and building knowledge graphs, building ontologies for diseases, and then identifying some potential targets, repurposing opportunities, and based on that, suggesting possible hits. And so some of the very well-known and widely advertised use cases from like last year, where deep learning type of algorithms were used to actually advance science in the pharmaceutical research, were, for example, DeepMind's AlphaFold, which basically allowed to predict protein 3D shape from the amino acid sequence, and that was the very fairly complex problem. And another example is in silico medicines, GAN infrastructure, generative adversarial network-based system, which basically was able to suggest quite potent kinase inhibitor within like days. And a lot of other examples where other types of molecules, like, for example, oligonucleotide molecules, were suggested by the deep neural net. And again, it was primarily involving automatic feature learning and data representation of chemical and biological data, which is important here. Another important aspect of applying deep learning is that it can basically generalize to different types of data and generalize over different data sets. And with that, it can be used not only specifically for hit discovery, but for target identification, let's say based on the publicly available unstructured data from like text, from articles, research articles, like patents, and also from structured data like bioactivity measurements or compound collections. So all this knowledge becomes the ontology on which some virtual screening campaigns can be basically then developed. And this is kind of new wave in the computational heat discovery. So yeah, much as the deep learning useful for all these types of applications, there is one problem with computational methods in general. Basically, computers tend to suggest molecules that are kind of hard to synthesize sometimes, and sometimes the synthesis step and validation step becomes kind of bottleneck for large-scale virtual screening campaigns, etc. And so here where Enamine's real technologies becomes very useful, and essentially the idea, as was already a little bit mentioned by the speakers today, but the idea that Enamine has a stock of qualified reagents, building blocks, and also a set of well-validated, established reactions. And so this gives a matrix for essentially creating the space of lead-like compounds, which are synthetically accessible, meaning that they can be fairly quickly synthesized and delivered. And so if the computational strategy is applied, like for example, virtual screening is applied on this data set, then it is much less risk involved that the compounds are poorly synthesizable, let's say. So this is the strategy to minimize the risk of synthesizability, and considering that the real space now is as large as around 30 billion molecules, it means that large-scale computational experiments can be run on this data, and having this backup by Enamine that compounds can actually be synthesized, rapidly tested, and validated in the assays, relevant assays. So Enamine is partnering with a fairly long list of drug discovery companies or computational developers, software developers. And one interesting use case is the partnership with Atomwise, where in 2019, I guess, where a large-scale screening campaign on 10 billion compounds was conducted. And the interesting part about Atomwise is that they use so-called convolutional neural network, AtomNet, which they essentially the idea is that like an image recognition, for example, the network first sees the image as generally a number of pixels with different color attributes, and then it basically starts convoluting it and basically understanding first the different color regions, then edges, then shapes, and then the whole objects, and finally it can basically understand what is written in the image, what is, for example, a dog, an image, a dog, a blue sky, you know, a grass, a forest, etc. So we have the output, the actual prediction of what is in the image. Similar strategy can be applied with actual chemical and biological data, where, for example, the protein pocket can be kind of represented numerically, and then the molecular structures, of course, can also be represented numerically, and then this can be vectorized and basically fed into this convolutional neural net, so that the most optimal hits can be found in a very rapid manner and on a very large scale data. Like again, 10 billion compounds, something that is impossible to be modeled or screened by some other techniques, I guess. So the case study too was, I guess, in 2018, and the results were published in Nature. That was the collaboration with University of California, and there was the large-scale heat discovery involving 170 million make-on-demand compounds by Enamine. And then the result of that screening was essentially quite strong hits against several targets like AMPCB lactamase and D4 dopamine receptor. And the results of that particular research were a number of compounds, one of them being the most active inhibitor known. And then for the other target, a number of compounds with very high affinity has been identified following that screen. And of course, a lot of other insights has been uncovered. And again, the bottom line of this cooperation that Enamine can support this huge amount of molecules with actual synthesizability guarantee, so that whatever is suggested by the algorithm, the Enamine can support it with actual molecules within reasonable times, like 2-3 weeks or something like that. And then there was collaboration with Silico, which is a Canadian company, fairly strong developer of deep learning based platform for drug design. So the idea is that they can screen very large data sets of compounds against the whole proteome, human proteome, essentially many proteins. And then they can uncover not only one interaction, but actually unknown interactions between random molecules and random proteins. And then they can do it at scale. And not only they can get actual hits from that kind of screens, but they can actually elucidate some mechanisms of action, repurposing opportunities, and so on. And finally, the recent collaboration involved participation of Enamine in the COVID moonshot project, coordinated by a computational company, Postera. So the company basically has a machine learning based algorithm which can apply natural language processing type of models. Basically, it represents molecules as a set of characters and then applies algorithms similar to translation algorithms. And that way they can predict retrosynthetic schemes with high precision. And so the idea of the project was that a lot of suggestions of molecules have been received crowdsourced from different chemists around the globe, and then the algorithm helped to rapidly elucidate only very relevant molecules and predict optimal synthetic routes for those molecules, and also elucidate their potency and safety. So it was rapidly followed up by Enamine synthesis. And so the project is now well progressing towards some interesting hits against protease to tackle COVID pandemics. So summarizing everything that I have mentioned is that we are now experiencing the confluence of technologies where essentially machine learning and deep learning are becoming enabled by new, first of all, hardware, highly powerful hardware, new kind of chips specifically for machine learning computations. They are very different from usual computers that we are using. And then by scalable cloud computing infrastructures. Those two things, they are becoming enabling factors for large-scale deep learning models to be actually feasible. And then comes the Enamine real space, which is the only available and the largest space on the market, which can be used by machine learning practitioners or computational chemists or companies to basically safely explore new ideas and be sure that they are actually supported by real chemical guarantee that the molecules will be delivered for the assay validation and other experiments.